Yeah, good morning, ladies and gentlemen. At first, I'd like to thank Rajiv very much for the invitation to give a talk here on genomics-based marker development and breeding for resistance in barley. But before coming to marker development and gene isolation, just a short introduction into barley. Barley is an inbreeding species with 7 chromosomes and a genome size of around 5.3 gigabase. In Germany, it's the third most important crop species, and in Europe, it's grown on around 24 million hectares. On the worldwide level, it's the fourth most important species with 51 million hectares, but in India, it's of minor importance with only 780,000 hectares. Barley is mainly used for animal feed, but also for human nutrition, especially of man as beer, but it's also gaining rising importance as a direct diet because of its high beta-glucan content, which is supposed to lower the blood cholesterol level. So, during the last decades, barley yields, for example, in Germany, have more than doubled, now being around 7 tons per hectare and year, while it's on the worldwide level on around 4 tons per hectare. However, each year, severe yield losses are caused in barley by fungal and viral pathogens. For example, leaf rust, stripe rust, mildew, net blotch, scald, barley mite, barley yellow mosaic virus, and barley yellow dwarf virus. Therefore, breeding for resistance to avoid these yield losses and to contribute to a consumer and environmentally friendly barley production is of prime importance. Due to this, soon after the first molecular map has been developed by Andreas Graner, who gave his talk yesterday already, attempts were conducted to develop molecular markers. And besides an innumerable amount of QTLs, which I will not talk about today, more than 80 genes against these pathogens, mainly derived from genetic resources, have been tagged in the barley genome. So, respective markers now allow efficient marker-based selection, efficient marker-based back-crossing procedures needed for incorporating of these resistant genes derived from exotic germplasm mainly, which means foreground selection by these markers and background selection by high-throughput marker systems available today. But these respective markers also allow pyramiding of resistance genes, which may lead to more durable and long-lasting resistance or more effective resistance. And one example for this is tolerance to barley yellow dwarf virus. Barley yellow dwarf virus is an aphid-transmitted virus and on the worldwide level the most important viral disease of cereals. So, for tolerance to this viral diseases, besides other 3 loci are known, which is RYD2 on chromosome 3H, RYD3 on chromosome 6H, and the QTL on chromosome 2H. So by respective markers and using DH lines, all allele combinations were developed. And for this, molecular markers are essential tools because on the phenotypic level, this combination cannot be distinguished by this combination. And now to make a long story short, you see the results here. On the left-hand side, you see a line carrying all negative alleles at these 3 loci after artificial infection, while on the right side, you see a line carrying all positive alleles. And what's more important now, I've talked about tolerance, but if we combine RYD2 and RYD3, we see that there's a considerable reduction of the virus titer, which means combination of these alleles led to quantitative resistance instead of tolerance. So, developing such molecular markers long time ago was a very time-consuming process. And the older ones among us may remember techniques like RFLPs, rapid genomic SSRs, and AFLPs. Today in barley, we have medium to high throughput molecular techniques available, like the DARTs Anjay told about yesterday, then different iSelect chips, the 9K iSelect chip, a 50K chip is supposed to become soon, then genotyping by sequencing, we heard about this, exome capture, and besides this, in Barley, the so-called genome zipper is available, which is a virtual order based on synteny to Brachypodium, rice, sorghum, and sequence information in Barley. So a virtual order of the genes available in Barley. Besides this, a physical, genetic, and functional assembly has been published already. And now to make these markers developed from these techniques available, besides the Caspar assay, which was mentioned several times already, we have developed a cost-effective pyrosequencing approach and a CAPS approach for multiplexing for using these markers for foreground selection. So these medium to high throughput marker techniques allow, as we heard already, the detection of QTL for resistances using association genetic studies. And I will not talk about this very much, but you see here in a set of German winter barley cultivars and phenotype for powdery mildew resistance at 12 locations over 3 years, we found several marker trait associations involved in powdery mildew resistance, and for example, most likely the one here on chromosome 4H is the MLG locus. But these markers or these techniques are also valuable tools for marker development in biparental populations in order to reduce linkage drag and to make these, let's say, resistant genes accessible for introgression into high-yielding adapted cultivars. And on the worldwide level, one of the most important barley pathogens is leaf rust. And with the exception of RPH7, RPH15, and RPH16, all the resistant genes known so far have been overcome by several isolates. So long time ago, we started a screening program and identified a Serbian landrace called MBR1012, which turned out to be resistant against the most aggressive strain of leaf rust known in Europe. And so in the first step, we mapped this gene in around 100 DH populations using conventional markers on the short arm of chromosome 1H in an interval of around 6 centimorgan. So at that time, the 9Ki select chip came up, and we used this population for developing our own map, and you see by this approach we were able to identify 311 markers on chromosome 1H, and within our target interval 16 additional markers were mapped. And so in the next step, Dragan Perovic in my group took our map, took the map of Jordi Comadran, which was published in 2012, and with the collaboration of Jeffrey Endelman's group using the software Dagger, he constructed a consensus map, and in the end, 50 potential markers were mapped in this interval. So some of these markers were then used to be mapped in a medium resolution mapping population going back to around 1,500 F2 lines, which were developed into segmental rills. And these markers were also assigned to the genome zipper, and then we used these markers developed from the genome zipper and the iSelect markers, and in the end we were able to narrow down the region to around 0.2 centimorgan. So now this gene can be introduced into high-yielding barley cultivars by these markers with a reduced linkage drag. And this also works for assessing the secondary gene pool of barley. The secondary gene pool of barley only comprises of Hordeum bulbosum. And in collaboration with Richard Pickering group at that time in New Zealand, we identified an Hordeum bulbosum introgression line carrying introgression on the long arm of chromosome 2HL, being tolerant against barley yellow dwarf virus. So in a first step, we crossed this line to an adapted cultivar, developed DH lines. It turned out that half of these lines carried the introgression, and then we used 9 markers to identify recombinations within this introgression. And for good luck, we were able to identify 27 different recombinations in this introgression. So again, we analyzed these lines with the 9Ki select chip. We developed additional markers by using the genome SIBR, and in the end, we were able to narrow down this fragment from around 25 centimorgan to around 3 centimorgan, and this fragment is carrying the Barley yellow dwarf virus tolerance. So in the next step, we assigned these markers to the physical map of Barley in order to get more detailed information, what genes are there, and we constructed a non-gridded bug library in Hélène Berger's lab at Toulouse from this introgression line. And actually, this bug library is screened with these markers in order to identify a bug contig and to identify candidate genes. We did not heard about much during the conference up to now, but in collaboration with the IPK, we also conducted RNA-seq, and we identified at the end 12 genes downregulated in the line carrying the introgression, and all these genes are located on chromosome 2HL, and they are carrying SNPs. So these are now under detailed analysis. So using these techniques, we were also able to narrow down this introgression fragment so that it now may be used in barley breeding. So, in the last part of my talk, I will now talk about gene isolation using these techniques, and I will do this with respect to barley mild, barley yellow mosaic virus, and therefore just a short introduction into barley yellow mosaic virus disease. Barley mild, barley yellow mosaic virus was first detected in Japan in the 1940s, and in Europe it was first found in 1978 in Germany. And since that time, it has become one of the most important diseases of winter barley, due to a constant spread of the area infested, due to the appearance of new resistance-breaking virus strains, and especially due to yield losses, which can be up to 40 to 50%, and which cannot be prevented by spraying chemicals, because these viruses are transmitted by the soil-borne Plasmodiophorid Polymyxa graminis. So, as already mentioned, after the first molecular maps have become available, special attempts were conducted to map resistance genes against barley mild, barley yellow mosaic virus. And as you can see here, several loci derived from Hordeum vulgare have been mapped, and some too derived from Hordeum bulbosum. So, already in 1993, we started a map-based isolation of the RYM number 4, RYM number 5 locus. And for map-based gene isolation, also in the area of genomics, if you are dealing with a quite complex genome and a phenotype which cannot be accessed in F2 plants, you have to develop a high-resolution mapping population, which means to identify segmental homozygous recombinants, which then can be used for marker saturation and for repeated phenotyping. And for this, we analyzed large F2 populations comprising of around 5,000 F2s, which is equivalent to a resolution of 0.01 centimorgans with flanking markers, in order to identify homozygous recombinant plants and heterozygous recombinant plants, because only these are informative with respect to mapping new markers. The heterozygous ones were then selved, and then the 3 homozygous recombinants were identified, and these homozygous recombinants can then be used for repeated, this means reliable phenotyping, for resistance to these viruses and for marker saturation. And with this, we start in collaboration with Andreas Graner in 1993. And in the end, we succeed to isolate the RYM number 4, RYM number 5 locus 12 years later, which means in 2005. And one reason for this is that marker saturation at that time, it was just trial and error. For example, for marker saturation of this area, we analyzed around 2,048 AFLP primer combinations and 1,200 rapids. These correspond to around 200,000 loci, and at the end, we were able to map 7 additional markers within this interval, and we were not able to bridge the locus. So let's say in a mood of desperation, at the end, we sequenced a back contig, and for good luck, it turned out that in this back contig, only 2 genes are present. And one of these genes was a translation initiation factor 4E, which at that time has been identified as a virus resistance gene in other pathosystems. And in the end, we were able to show that resistance at the RYM number 4, RYM number 5 locus is due to mutations in this translocation initiation factor. So today, this procedure, which took 12 years, can be designed much more efficient. As already mentioned, you still have to use high-resolution mapping populations to facilitate efficient phenotyping. But there are high-density maps available in Barley, so you can anchor your flanking markers to these maps and just use the markers known here inside for marker saturation. Then you can anchor the closer ones to the genome zipper, identify additional markers here, use genotyping by sequencing or exome capture, at least to identify candidate genes, which had then been proven by transformation, by tilling, or by RNAe. And this approach we used for the isolation of the RYM number 11 locus located on chromosome 4HL. Again, a high-resolution mapping population was constructed, and by using the genome zipper, we identified one marker co-segregating with RYM number 11. And so this marker then was analyzed in detail, and it turned out that this is due to a rather large deletion in a gene coding for a protein disulfide isomerase. And next, Ping Yang and the group of Niels Stein proved that this is really the gene which encodes for the RYM number 11 locus. He used therefore tilling of EMS-induced mutants, test for allelism with naturally occurring RYM number 1 alleles, and he used the transgene-induced complementation, and the results you see here. Resistance is inherited in a recessive manner, so resistant plants were transformed with the functional protein disulfide isomerase gene, and as you can see here, plants turned out to be susceptible. So once you have these genes identified, you can efficiently conduct allele mining in large gene bank collections, and Ping analyzed around 1,800 accessions, and by this he identified 4 different naturally occurring alleles of this protein disulfide isomerase, and all of the alleles code for non-functional variants of this gene. So besides this, he identified 2 additional alleles in the EMS mutant collection, and in the next step, he developed allele-specific markers for these alleles. And now, as we already had done allele mining for the RYM number 4, RYM number 5 locus as well, and around 10 alleles of this locus were detected, now these alleles can be combined efficiently by these markers. And as these genes are interacting in different stages of the virus developmental life cycle, it may be supposed that by combining these alleles, more durable resistance may be created. And as already shown, several markers have been developed, or several genes have been mapped for resistance to these diseases. And so at present, we also conduct isolation of the RYM number 7 locus, which is exclusively efficient against Barley mild mosaic virus. And here we used a different approach. In a very small DH population, we first mapped this gene, and we conducted marker saturation using the genome zipper. At present, a high-resolution mapping population is in progress where these genes are mapped in order to isolate this gene. Furthermore, just very recently, by the analysis of this gene bank accession, we mapped another gene on chromosome 5H. And besides this, we identified in this genetic resource an allele of the RYM number 4, RYM number 5 locus, which is efficient only against Barley mild mosaic virus. This means in these genetic resources, 2 genes had been incorporated or pyramided already. And so we just conducted the same procedure, and up to now, we were able to map this gene to an interval of around 0.3 centimorgan, and the parental lines of this population are at the moment analyzed by exome capture in order to identify additional markers and to isolate this Barley mild, Barley yellow mosaic virus gene in cooperation with Andreas Grana. So, up to now, we were quite lucky with marker development and with isolation of these resistance genes, but a different story is RYM number 13. Again, we constructed a high-resolution mapping population, developed markers, and then we got a gap here, where we found a lot of double recombinations and no additional marker. And when we got first phenotypic data of the segmental recombinant inbred lines, it turned out that our gene is exactly mapped within this region. So, in the first step, to prove that this mapping was correct here and that our gene is not lying outside our interval, we conducted genotyping by sequencing of around 60 lines of these DH population, and we have done genotyping by sequencing of around 50 lines of these recombinant inbred lines. And by this, as we only find, let's say, significant deviations in allele frequency at exactly this region of chromosome 4HA, we were sure that our gene is really located within this region. By genotyping by sequencing, we've identified additional SNPs, anchored these to the PopSec mapping population developed by Martin Mascher, and assigned these SNPs to already sequenced fingerprint contacts derived from sequencing the minimum tiling path Andreas mentioned yesterday. And there we identified a contact which is located here carrying 22 genes. And one of these genes may be a candidate gene for this RYM number 13 locus, and this we are at the moment analyzing in much detail. So, coming to an end now, I hope I could show you that the availability of dense marker maps, high-throughput genotyping platforms, physical maps and genome sequences of Barley itself and related species facilitates an enhanced marker development and isolation of resistance genes, thereby leading to a deeper understanding of resistance and the transfer of marker-based selection to the allele level. This will lead to a more directed use of plant genetic resources in Barley breeding by minimizing linkage drag, thereby broadening the genetic base of resistance. So the work I have presented here has been the work of many, many people in my working group, and here I want to mention especially Dragan Perovic, who is involved in all these molecular work. Then Thomas Lübken, who is involved in the isolation of RYM number 13. Sandra Lehmann and Katja Perner, who are doing the isolation of the other resistant genes. I want to especially thank Andreas Graner and Niels Stein for a long-lasting, fruitful collaboration, and Ping Yang, who has done the isolation of RYM number 13. At least these organizations for funding all of this research, and you for your attention. Thank you very much.
Thank you very much for your interesting presentation. Are there questions? No question on Barley?
Not a crop of prime importance.
Well, yeah, that was... well, perhaps I would like just to, more out of curiosity, I've been a barley breeder for 25 years, so I very recently moved out of barley. You definitely demonstrated the much faster identification of genes, markers. Do you have any case where those have been, using the most recent techniques, transfer and varieties developed, or work in progress?
Yeah, let's say I've talked about these pyramided barley yellow dwarf genes, and let's say this has been directly implemented into German breeding companies, and the first lines which have been developed out of these lines are now in official testing. And so this is a good example for transferring, let's say, genes with markers into adapted breeding material. And on the other hand, let's say these plant breeders are very interested in these barley mild, barley yellow mosaic genes, because that's a knockout criteria for most parts of Germany. Barley varieties have to be resistant to these lines, and now let's say if they have a gene or a allele specific marker like for RYM number 11, this will be used in barley breeding.
Thank you. And in the meantime, there is a question.
First, thank you very much for your introduction. That's very excellent work. Several cases you mentioned you use high density markers, and also there's one or two cases basically you step by step increase the density of the markers. So which molecular marker system are you using? Array or sequencing or other?
Different way. Let's say when we want to saturate a region for gene isolation, we try to do this by using all the information available from high density maps and from the genome zipper. So we develop specific markers for this based on already known knowledge. But let's say in case genotyping by sequences is very cheap, the 9Ki select things are getting cheaper and cheaper. So it may be another option I did not talk about, just to analyze recombinant inbred lines using these high densities.
Thank you.
With that form.
On the resistance breakdown, actually, in this virus, resistance breakdown.
Resistance breakdown in these viruses.
If it is the case, how these markers will going to perform? Because in other crops, developing markers for viral resistance is very difficult, actually.
Yes.
So viral genome is quite mutative in such situations.
Let's say for RYM number 11, this gene, the protein disulfide isomerases are involved in correct folding of genes. So the transport or the correct assembly of these viruses may be prohibited due to the non-function of this gene. So I think this is quite a durable thing. For RYM number 4, RYM number 5, it's that the viruses have a VPG which resembles the cap structure of the mRNA, and this binds to the to this translation initiation factor. And so just a few mutations in this VPG maybe lead to resistance breakdown. Therefore, let's say just a feeling, no proof, I think that the RYM number 11 locus, which codes for a non-functional gene, may be longer lasting. I hope.
Okay, thank you. If there are no more questions, I thank Frank, and we move now to the next presentation.